Hi everyone, my name is Motika Betran. So today we are going to be looking at the last part of carbohydrates, which is polysaccharides. So what exactly are polysaccharides? So polysaccharides, they are a group of carbohydrates that consist of several monosaccharides that are linked together by a special type of bonds known as the O-glycosidic bonds. And these polysaccharides, they play important role in animals such as energy storage, structure, and also protection. So these polysaccharides, they are broadly classified into two different categories. We have the homopolysaccharides and we have the heteropolysaccharides. The homopolysaccharides are those type of polysaccharides that are made up of the same type of monomers which are repeating and they are connected together by the O-glycosidic bonds. While the heteropolysaccharides, they are those type of polysaccharides that are made up of different monosaccharides which are also connected by the O-glycosidic bonds. But for the purpose of this video, we are going to focus mainly on the homopolysaccharides, and we are going to see three different homopolysaccharides here, which is glycogen, we have starch, and also cellulose. So we are going to begin with glycogen. So glycogen is the most common polysaccharide that is found in animals. So animals, they store their excess carbohydrate, they store their excess glucose in the form of glycogen. So glycogen is a homopolysaccharide that is made up of repeating glucose residues which are connected by the alpha-1,4 glycosidic bonds. And we have branch points. So after every 8 to 12 glucose residues, we have a branch point, which is the alpha-1,6 glycosidic bond. And this makes glycogen to be highly branched. So we have the alpha-1,4 glycosidic bond, alpha because the bond is pointing down, which is connecting the glucose residues together. And then after every 8 to 12 glucose residues, we have a branch point, which is the alpha-1,6 glycosidic bond. Now the second type of homopolysaccharide that we are going to see is starch, which exists in plants as insoluble starch granules in the chloroplast. And each starch granule consists of a mixture of two polysaccharides, that is amylose and amylopectin. So amylose and amylopectin, they are the two polysaccharide forms of starch. Amylose is long and unbranched, and it is made up of glucose residues that are connected by the alpha-1,4 glycosidic bonds, just like the linear form here, while amylopectin is highly branched form of starch. So amylopectin is a highly branched form of starch, and branching occurs in amylopectin at every 24 to 30 glucose residues, meaning that in the linear form, every 24 to 30 glucose residues, you are going to see a branch point, just like in glycogen here. So if you look at the structure of starch here, particularly amylopectin, if you look at the structure of amylopectin, after every 22 to 24 to 30 glucose residues, you have this kind of branch point, the alpha-1,6 glycosidic bond, just like in glycogen. While amylose is linear and unbranched. So in amylose, you just have a series of glucose residues that are connected by alpha-1,4 glycosidic bonds, and there's no branch point there. So now, what's the difference now between amylopectin and glycogen? You discover that in glycogen, branching is more frequent. I earlier said that in glycogen residues, you have a branch point, while in amylopectin, the branching occurs at every 24 to 30 glucose residues, which makes branching in glycogen to be more frequent compared to that one in amylopectin, which is a form of starch. Now, we come now to cellulose. In cellulose, you discover that what? It is a fibrous, tough, water-insoluble substance found in plant cell walls, and it is unbranched linear homopolysaccharide. So cellulose is unbranched, it is linear, and it is a homopolysaccharide, meaning that it is made up of the same monomers, the same monosaccharide, specifically glucose. So cellulose is the same, just like amylose, but the only difference between the two is that what? Amylose have the alpha-1,4 glycosidic bonds, while cellulose have but the beta-1,4 glycosidic bonds as seen on this structure here. So in cellulose, you have repeating glucose residues, right, which are connected by the beta because the bond from this anomeric hydroxyl group of the first glucose residue is pointing up. So this is the kind of bond that is formed there, which is the beta-1,4 glycosidic bond. So the beta-1,4, indicating that the hydroxyl group is coming from the anomeric carbon of these first glucose residues, and the hydroxyl group of carbon 4 of the next glucose residues to give you this beta-1,4 glycosidic bond. So it is a linear molecule. There's no branching in cellulose, and the type of bonds that are found in cellulose is beta 1,4 glycosidic bonds. So the only difference between cellulose and amylose is that amylose have alpha 1,4 glycosidic bond, while cellulose have the beta 1,4 glycosidic bonds. Now, these are not all about the homopolysaccharides that we have. There's also another one known as dextran. So dextran is also a homopolysaccharide which is mostly found in bacteria and yeast. And the kind of bonds that are found in that kind of homopolysaccharide, they are the alpha 1,6 glycosidic bonds. And we also have brand points that occur at alpha 1,2 and alpha 1,3 depending on the type of bacteria or the type of yeast. So if you liked our video, please comment, share, and also subscribe to our YouTube channel. Thank you very much and see you in our next lecture.